This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. You're listening to TWiM, This Week in Microbiology, episode 167, recorded on December 14th, 2017. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello.
Hey, we got snow here, Michele.
Ooh, so do we. A lot of it. I think we got maybe 5 inches yesterday. A lot of the schools are closed today.
Wow.
It's a winter wonderland.
We had 3 inches on Saturday. It melted, and then last night it did another 1 or 2 inches, and it seems to be melting. Although it's very cold here now.
December. It is December.
Yeah.
So,
of course, in certain parts of the country, it's not snowing. Also joining us from Charleston, South Carolina, Michael Schmidt.
Well, it did snow in the upstate of South Carolina this past weekend. And so, but we are fortunate here on the coast. It's just a cold, damp rain. And today it's a little bit sunny. I had to look out the window, and it's a little bit sunny, but it is December. And but I think snow is a better alternative than the poor souls out west dealing with the fires.
Yep, fires.
I mean, those poor souls in Los Angeles and Santa Barbara and Ventura County, it's just got to be, freaking everyone out.
Yep. Yeah, fires are tough. They can take your house. Snow.
Yeah, especially if the wind is blowing at 60 miles an hour. Yeah, it's just unpredictable.
We will be without Elio for the next 2 episodes because he's actually out east on vacation with his family.
Nice.
So enjoy that, Elio. And in the meanwhile, we will continue to deliver to you the finest microbiology that a free podcast can deliver.
At least we're going to try.
And since we are all card-carrying microbiologists, you'll probably do all right.
Yes.
All right. We have 2 papers. We have a snippet and a paper. And for the second episode in a row, we're going to talk about influenza virus. Michele picked this paper.
I did. It's titled Pandemic H1N1 Influenza A Viruses Suppress Immunogenic RIPK3-Driven Dendritic Cell Death. And it is by scientists from Mount Sinai's Medical School in New York City and the Fox Chase Cancer Center in Philadelphia. And the authors are Boris Hartman, Albrecht Zaslavsky, Nudelman, Pincus, Marjonovic, Schatzart, I'm sorry I'm butchering these names, Martinez Romero, Fenetria, Ingram, Ramos, Fernandez Sesma, Balajandran, Garcia Sastra, and Silfen.
Wow, that was a mouthful, huh?
Yeah, I'm sorry, that pronunciation's not my strong suit.
By the way, Mount Sinai Department Microbiology, that's where I got my PhD.
All right.
Many years ago. Yeah. No,
so
do you have an affinity for flu or RIPK3, Michele?
Well, it is flu season, and I thought this was a hopeful paper, actually. So H1N1, our listeners may have heard of this. It is the type of flu virus that caused the famous pandemic in 1918 that is estimated to have killed 50 million people worldwide. Another version came through in 2009. You might remember the swine flu that caused a lot of sickness and death. So what are these names? Where do they come from? The H and the N both refer to surface proteins on the viral shell. The H is hemagglutinin, N is neuraminidase. And we have to name the viruses because flu is a quick change artist. It has genetic mechanisms to alter at a very high rate its surface structures so that the virus that we see this winter will be completely replaced or mostly replaced by another virus variant next year. And that's why we have to be diligent and go out and get our flu vaccines. Have you two been vaccinated this year?
Yes, I have.
All right.
I have too.
And how did they do with predicting the vaccine for this year's viruses, Vincent? Do you know?
Well, one of the strains is the vaccine has an H3N2 and H1N1. and one or two influenza B strains.
Mm-hmm.
The H1N1 is a good match, but H3N2 is a problem because, as we discussed last time, although the strain was matched, when you grow it in eggs, which is what you do for most of the vaccine, you lose the mutation that you need for a good match. And so it's not a good match. However, you'll still have less serious disease, and the other isolates will protect you. So it's good to get it. I was listening to a podcast, a tech podcast, a couple of weeks ago, and they were talking about this mismatch, and they were lamenting that it wouldn't be worth getting the vaccine. So I wrote into them, and I said, it's still worth getting because you'll get less serious disease, and these other two strains are still well matched. And you know what they did? They made fun of me and said I was a big pharma shill just wanting to sell vaccines.
Oh my.
That's awful.
I was stunned. Here I was trying to just inform them, right? So you don't have to get the flu vaccine, but you should make a decision based on good information. And they made fun of me. And the thing is, I don't even work for Big Pharma.
Yeah, right. We work for free. No. Plus, isn't it money in the bank? Even if it's not a perfect match, we now have that information in our memory cells. And so next year, we'll have protection if that other one arises.
In a way, yeah, it depends. I mean, the memory for flu vaccines isn't great, but
I see.
Yes. But even if it's not that, even if there's a mismatch, you'll get infected, but you'll have less serious disease.
Right. It's not a black and white phenomenon.
Exactly.
Right?
Exactly.
Mm-hmm. Yeah. So the question arises, how is it that seasonal flu will kill people, especially the very young, the very old, people that are otherwise ill? But by and large, people can recover from seasonal flu. And yet we have these pandemic versions of the H1N1 that wiped out millions of people. So this group wants to understand really at a mechanistic level, what is the difference between the seasonal flu and the dreaded pandemic H1N1s. So the system that they use is white blood cells called dendritic cells from human blood. They do all their studies with human cells because the components that recognize and fight infection are somewhat different between mice and humans. So that's one strength of this paper. And what they know from the literature is that we have in our bodies surveillance systems that are able to detect and quickly respond to foreign molecules made by microbes, including viral RNA. So in this case, we know that if your cells in the lung are infected with flu virus, the cells can detect the viral RNA through the surveillance pathway, and that infected dendritic cell, in this case, will recognize that it's infected and then purposely undergo a programmed cell death. And by doing so, they deny the virus a chance to replicate, because of course viruses depend on host machinery to make copies of themselves. And they also send out signals. So they don't just die quietly, but they scream out to the neighboring white blood cells, I'm infected, and they capture key information about who has infected them, as though they've taken a snapshot and have shown it to the rest of the immune system. So this is called antigen presentation. So when the infected dendritic cell dies, undergoes this suicidal programmed death, it releases signals and it provides captured antigen to neighboring uninfected dendritic cells, which then take that information and trigger T cells, CD8 T cells, to begin to multiply. And those T cells are then responsible for moving around the body, finding infected cells and killing them. So that's why most of us will be miserable if we get the flu because we didn't get our shots, but we will recover. So then the question is,
you'll recover more quickly.
We'll recover more quickly. Yeah, you'll still perhaps wish you could die because you're so sick, but you won't. So how is it that this pandemic flu virus is different, is the question they asked. So in earlier work, they had identified a signal transduction pathway that's important for this, and one of the key components is called, in the title, it's the RIPK3. So this is a receptor what does the I stand for? Receptor interacting protein kinase, but I'm going to call it the rest in peace kinase to remind us.
So you remember what it's doing.
Yeah, that it's part of this coordinated plan to die and at the same time capture the antigen and activate my neighboring white blood cells to fight the infection. So what they do in the first figure 1, is they have these dendritic cells and they infect them with either a strain of the seasonal flu or the pandemic flu. And then they measure cell death. And they do that in a couple ways. You can look at the morphology of the cell. When they undergo this programmed death, their nuclei become all fragmented, so you can measure that. They also measured how many fragmented DNA molecules were inside the dendritic cells, and they could also measure how much components from the cytoplasm were released as these cells burst. And what we see very clearly is that the seasonal flu viruses do trigger this programmed cell death, whereas the pandemic viruses trigger less of it. So only about 10% of the infected cells exposed to the pandemic virus die, whereas about 60% of the seasonal flu virus infected cells are undergoing this programmed death to alert their neighbors. So they know they have now an experimental system and they can use it to start to identify the mechanism. How are the cells recognizing and responding so quickly to this the seasonal flu virus, but not the pandemic virus. So in the next figure that I'll talk about, figure 2, they learned by using genetic tricks and drugs to block particular pathways, they were able to identify that RIPK3 was one of the key molecules in this host sensing pathway that coordinated the programmed cell death. And they also learned that the virus needed to replicate in order to cause this death. So then in figure 4, they did a really cool experiment where they first infected the dendritic cells with either the pandemic flu or the seasonal flu, and then they intentionally exposed it to synthetic RNA to give it a load of viral RNA. And by doing that, they learned that if the cell already had pandemic flu virus in it, it was now resistant to a stimulus of synthetic RNA. So in that experiment, then they could deduce that the pandemic flu actually could inhibit the signaling triggered intentionally by a synthetic RNA molecule. So now they can start to think about mechanisms. Somehow it's inhibiting this recognition and cell death pathway. So then I'll jump to figure 6b, which I found really elegant. At the whole figure, what they do is take advantage of the fact that flu viruses are segmented. So they have 8 different chromosomes, if you will. And it's possible experimentally then to make viruses that are chimeras that have, 7 segments from the pandemic virus and only 1 segment from the seasonal virus and vice versa. So they made a whole series of these chimeras and then could ask which chromosomal segment did the suppression of cell death track with? What did it map to? And by doing that, they were able to determine that it is the hemagglutinin molecule, one of the surface protein molecules, the H in the H1N1, that was contributing and accounting for the ability to block programmed cell death. To explore that further, they did a really, I thought, intelligent experiment, which is they took advantage of many different H1N1 viruses that have been circulating in the human population over decades. And so they looked at, I think, 8 different viruses from different time periods. So they would have slightly different versions of the H protein on their surface. And they sequenced them and asked how similar are each of these H proteins to the pandemic potent H protein that can inhibit cell death. And they then directly measured cell death inhibition by each of those variants. And in doing so, they could come up with genetic data from human populations that was consistent with their model, that in fact it is this H hemagglutinin protein on the surface that correlates with blocking cell death. Is that clear? How am I doing so
very
far?
Very clear. Beautiful.
Okay, good. Yeah, I thought that was a really great experiment. I'm curious, Vincent, how many different types of flu genomes are out there for experimentalists to
thousands.
Really?
There are thousands and thousands and thousands, because they do a lot. WHO coordinates global surveillance, and they have tons of sequences in the data bank, and you can just get those and synthesize the segments and put together your own virus.
Wow.
Very easy to do. You make, if you take 8 plasmids, one for each segment, and you put them in cell, you can get virus out. So you could make any combination of all these sequences that you want.
Wow. So it's like nature has done the experiment.
This is actually then illustrating the hallmark of this virus, namely the shift and drift. So as in the pandemic variant, you have not yet undergone the sin of all RNA viruses, namely the drift in its genome, because the RNA viruses, especially flu, has no proofreading activity. And so as with each round of replication, there is some drift in this hemagglutination antigen. And at the same time, you have the ability of the influenza virus to pick up segments from other viruses, or that's the antigenic shift. And so, the thing that troubled me is what's unique about this hemagglutinin antigen in the pandemic strain. How does the dendritic cell effectively know that this is the pandemic strain? And that's their elegant experiment with the mixing and the matching.
They then did another experiment that is going to help them think about mechanism, which was they asked if they could just purify the hemagglutinin protein and add that to the dendritic cells. Would that be sufficient to inhibit this cell death pathway? And based on that and some other really careful experiments, they were able to deduce that only when, they are predicting that, in fact, the infected cell has to be newly synthesizing the hemagglutinin RNA or protein in order for it to be competent to block this surveillance pathway, the host surveillance pathway. So it's not going to be as simple as just adding HA protein to a cell and have it processed. So they're starting to think about what might actually be happening during viral replication to have this occur. But that gets maybe into a little more nuance. I do want to describe one other experiment that I thought was really beautiful. They did verify that a dendritic cell that was infected with the seasonal virus was able to not only commit this programmed cell death, but those infected cells did in fact present antigen to other dendritic cells, which could then stimulate proliferation of the appropriate killer T cells that would then go do the job, the effector job of hunting down and killing infected host cells. So they verified that T cell proliferation was also occurring for the seasonal virus, but not for the pandemic virus. So again, to this group's credit, they wanted to take advantage of whatever data was out there and ask whether this was likely to be happening in an intact human with their whole immune system. And so they took advantage of a published data set. This was work that was published in PLOS Pathogens in 2015 by another group who had enrolled 1,600 healthy adults and then followed those adults during the flu season. And they took blood samples and profiled their white blood cell gene expression while they were healthy. And then when a subset of those people in this trial or experiment came down with the flu, they were able then to take another blood sample early in the infection and then a few days after their flu infection, and again, capture the RNA profile of their white blood cells. So that data is publicly available, and what this group did was use computational methods to extract from that data set information about what genes were expressed in the healthy people before they were infected and then after they were infected, either with the seasonal virus or the pandemic virus. And specifically, what they did was pull out molecular signatures of the T cell population, because their earlier experiments had shown that the seasonal virus does trigger lots of T cells to expand, whereas the pandemic virus does not. And so in figure 7E, they show data from 9 different patients in each of those groups. And again, statistically, they do find that there are fewer T cells in the blood of a human who was infected with an H1N1 of the pandemic type. So lower T cells, in fact, than they measured in the control or the comparable group that had a seasonal flu. So, they admit that this was only 9 patients and it's somewhat indirect, but nevertheless, it is consistent with their model that they developed from these experiments I've described and many others that I don't have time to describe, that in fact, the pandemic viruses have got a special hemagglutinin on their surface that when the virus is replicating and making these new HA molecules, in some way, they're interfering with the natural surveillance system of our white blood cells that typically can recognize the viral RNA and trigger programmed cell death, alert their neighbors of the problem. But the pandemic strain is able to somehow short circuit that pathway. And so as a result, the immune system is not alerted and the virus then gets an upper hand. And presumably that's why you get a higher viral load and more illness and more death.
So what should we be vaccinating our people with to prevent a pandemic from occurring? Should we be looking at this HA molecule in greater detail to try to decipher what piece is unique? Or should we be focusing on the neuraminidase associated with the pandemic and make that more antigenically robust in order to affect sterilizing immunity for a vaccine candidate? Which way do you think we should go?
Well,
I don't know if there's an answer.
Yeah,
the main neutralizing antibodies are against the HA,
right,
not the NA. So we have to focus on the HA.
Mm-hmm.
I think it would be interesting to know if they can identify, what motif in the HA is responsible for this effect. And then you could do surveillance and say, are there any circulating strains that look like that and which we have to keep an eye on, right?
Yeah.
Because if there's some property of HA that makes it lethal like this in the mechanism suggested by this paper, then we should be able to track it. But I think in terms of a vaccine, the real thing is to make a universal flu vaccine, which will neutralize any strain out there. And, people are working on that.
Yeah, and they're avoiding the surface parts of the HA and the NA molecule, and instead going for the stalk, kind of the deeper embedded part of the molecule, which doesn't change as much from season to season. Exactly. So that's the strategy that's underway now. I think there are human trials underway.
Yeah, there are. Yeah.
Those. Yeah.
I guess in the next 10 years we'll have something licensed for that. That will be. I mean, even if it's not once in your life, if it's once every 10 years, that would help. But.
Right.
If that kind of vaccine could protect against, say, a brand new pandemic strain, then problem solved. And we could take all that flu research money and put it towards other stuff.
Yeah. I'm also wondering if knowledge of how this HA is interfering with this particular pro-inflammatory cell death, if there might be some clinical scenarios where you'd want to mimic that with maybe a small drug.
Yeah, it's a good idea.
To avoid over-amplification of the immune system, which can be, of course, toxic also.
I think that's a good idea that using a virus to probe these systems will give you some mechanistic insight that you can translate. Yeah, that's a good idea.
And drug development targets.
Yep.
Yeah. So,
or even for transplant.
Maybe.
To
could prevent rejection.
Right. Now, fortunately for us, we do have many different redundant pathways for detecting different invaders. Yeah. And so they will converge on a few molecules that maybe are the best drug target, but we might need a cocktail drug to block all of these cell death pathways.
This is a really cool paper, and it's in an open access journal, so listeners can go see it if they want.
They also did a great job of using color coding and schematics to make it easier to follow all their different constructs and chimeric viruses. So I appreciated that. It will be a good teaching paper for those of you who are looking for a neat way to teach some basic flu virology.
Right. Well, thank you, Michele. That was very nice.
Yeah.
It's a cool paper. Interesting stuff. And
Let's talk about 3D printing.
Okay. Yeah. I came across a paper in Science Advances, and it's called 3D Printing of Bacteria into Functional Complex Materials. So I saw the title and immediately said we have to do this, which could be dangerous because it may be that I don't understand any of the paper, but it's not the kind of paper that I would be fully comfortable with, but I think we can get the gist of it to you.
It's a really fascinating paper.
Yeah, the ideas are great. The authors are Manuel Schaffner, Roos, Coulter, Kilcher, and Studard, and they are from ETH Zurich and University College Dublin. So, as Elias would say, so what's going on here? The goal of this paper is to try and use the diversity of bacteria, which can live almost anywhere. They have huge, diverse metabolic activities, more so than any other organisms. They form biofilms, and these biofilms can adapt to their environment to match the conditions. They can degrade lots of compounds. They can form all sorts of chemicals to use this, and in fact, to use our ability to program all of these properties to create 3D materials that contain bacteria that can do things that are useful. They have two examples in this paper of useful things. One is to degrade a compound, and the other is to make what could be a medical device made out of bacterial cellulose.
The other thing you need to point out is what this really is a machine. It's a very, very small machine designed to do a specific task. And as Vincent said, task one that they demonstrate is to degrade a toxic waste product, and the product they're going after is phenol. And the other is to make a scaffold that they can grow skin on.
Yeah.
So it's absolutely fascinating. And the other thing that we always have to remember is biofilms need not be one single organism. And this is where the machine really gets pretty cool, because you can actually have organism one doing one particular task, then organism two doing a second task. And when you think about the three-dimensional printing, you can actually vary where the organisms are laid down in 3 dimensions.
Right.
So you can actually design your biofilm. It's going to be fun.
And use different colors of ink too, 2 or 3 different colors, so you could put different species of bacteria at different places. Yeah. It's really
so you know where they are.
Yeah. Exactly. So the idea here is to immobilize the bacteria in a hydrogel and try and manipulate it in a way to have beneficial properties. In addition to the 2 examples, they say we could make fuel cells, we could make biosensors this way by embedding bacteria and programming them to do certain things. So what they do in this paper is to create some proof of principle for this. They make it's a 3D printing platform. Now, if you think of a 3D printer, it lays down layers of, if you want to print, say, a virus particle, it would take the 3-dimensional coordinates, and then a machine would lay down layers, thin layers of a polymer, a plastic, or a clay-like material, which would then harden to form the final object. And what's going to happen here, they're going to embed bacteria into a matrix that will have strength and flexibility. And they call this functional living ink, or Flink.
I love that.
a biocompatible immobilization medium that exhibits the viscoelastic properties required for 3D printing through multi-material direct ink writing. So, that's complicated, but basically they have to make this matrix strong enough and printable. It has to be able to be squeezed through a nozzle and shaped into different forms and have different kinds of properties. And that's what they do in this paper. And they have
but
go ahead.
It's got to be have enough solidity that it will hold its form, but it also has to keep the bacteria alive.
That's right.
That's where the tension is in this design project.
Right.
Between those two goals.
So you want to put them in an ink that can be extruded, and what they do is they make filaments. Plus, you have to keep them alive, and they have to metabolize because you want to use that metabolic activity to produce something or to degrade something. And so what they've settled on is a hydrogel composed of hyaluronic acid, kappa-carrageenan, and fumed silica. And they've learned that this is compatible with bacterial growth and metabolism. It can be printed into scaffolds, and they go through a little bit of illustration to show that. And the first one is biodegradation by Pseudomonas putida, immobilized in a 3D printed lattice. And the second is making cellulose by A-xylinum. What's the A stand for? Michael must remember. Acetobacter.
Acetobacter. It's an acetogen. Yeah.
Makes cellulose. All right. So basically, they're going to put these bacteria in the inks, and then they're going to extrude these filaments into different shapes and see how they work. So they go through quite a bit of experimentation to find the right concentrations so the bacteria survive. They have to make sure these filaments don't dry out. They have to be strong enough so they don't shear. They have to flow properly out of the ink nozzle. I mean, these are all process-based issues that I don't usually think about with like shear forces on the printing process and all that sort of thing. They basically come up in the end with a good composition of this Flink. And they show that you can actually incorporate bacteria into very specific regions of a printed structure. They have a picture here with a shirt, a grid, and some other things. And you can see that the bacteria can be placed in very specific places. I'm not sure how that works, actually. I believe them, but I don't know how you would make sure the bacteria are in a particular place, right?
Just like you have a 4-color printing process, you can actually have the ink, because they're using my guess is a variation of the standard inkjet printer that we all know and love. Many of us have it in our office. And so you would put the bacteria in the black well or the yellow well or the cyan well, and it would just squirt it on when it's calling for black or blue or yellow.
Yeah, right. And just like printing. Yeah.
It's just like printing. And the trick, I think, is the matrix that they're using. It's nothing more than a grid pattern, and their inherent design of this grid pattern is that the bacteria will actually be able to grow out of the ink and actually fill the space.
Exactly.
And so that's what the cool part is that it's both solid and not. And so the microbes can actually get across or outside of the matrix that they're doing, very similar to the way biofilms actually continue to expand and slough and do all the wonders that biofilms do.
In fact, they even mentioned they loaded the tanks of printing ink with different bacteria. So it's exactly as Michael said. You can imagine wherever a certain color has to be, it would mix the inks together, and you get a mixture of your different bacteria. So they can be placed very specifically in the structure just by injecting them.
So, Vincent, imagine what this is going to do to your art competition in the future.
Oh, God.
Oh, yes, for sure.
Each bacteria could be a GFP or an RFP.
Yeah, sure. All different colors, yeah. And you can do, I mean, whoever's going to be able to afford this, right?
Right.
Even 3D printing is not cheap at the moment, I just went to a company online and ordered some 3D viruses printed. I have them here on the desk in front of me. They're about 2 inches in diameter, and they were $30 each, Mm-hmm. I could go broke on viruses at that price. But yeah,
you just have to go down to the dental school and meet your friends in the dental school. Many of the dentists are using 3D printers to print appliances that actually, night guards and other things. So they may have a 3D printer in their lab And if you get the coordinates of the virus, you can make your own. The materials aren't that expensive. What you're paying for is the hardware.
That's right.
And the know-how.
That's right.
And the know-how.
That's a beautiful setup for the second part of the paper, the cellulose producers.
That's right.
Right. So they have a lot of experiments where they have make sure the bacteria can live in these inks. They do viability studies. They do some cross-linking, look at viscosity and so forth. They do make a grid, which I don't know if you think of a screen with very large openings, they can print these bacteria in that kind of an orientation, and they look at the swelling of the gel in water versus medium, and then they do an experiment where they make one of these grids and they put Pseudomonas putida in it and ask, can it degrade phenol? So just think of like a screen, but much thicker and with bigger openings, and your bacteria are embedded in this. And you could place that, and they place this in a flask with phenol in it, and they show that the concentration decreases. And what they can do that's really cool, they can take out the grid, wash it, and put it back in again, and it will go through a second incubation period and remove even more phenol and convert it into biomass. And so they show that this actually works. Whether and a lot of bacteria are released from this grid, as Michael mentioned before. So it must be a combination of those plus the bacteria in the grid that are doing the degradation. So that's interesting. But Michael, tell me, why is it better than just if you have a phenol spill, can't you just inoculate your bacteria into the spill? Why is this better?
Well, if you think about it, a lot of times what you want to do is go into an aquifer. And what you will do is you will pump out the material, and then you can drive air across it, and you can vaporize. That's the way a lot of these aquifers are cleaned up, is they sink a well and they drive air across it and they vaporize it and let the volatile organic just diffuse into the atmosphere. So what you can imagine is that you can blow the phenol-laced air into this reactor technology, and the microbes are suspended in this very hydrophilic gel, and you're providing them a carbon source. And if they're bathed in the other essential materials that they need to grow, and they were using a minimal medium where the defined chemical composition is known, and the only carbon source that the microbe has access to is the phenol.
Right.
You can actually design it so it can degrade in real time. So rather than trying to figure out how to get the phenol out of the water or separate the water or concentrate the phenol, you can set up a surface to volume ratio so the air literally is feeding the microbes. And when the air comes out, the air is devoid of the phenol because you have that incredible driving force of the microbe wanting to eat needing to eat the carbon and energy source for it. This has actually got so many applications in the environmental world. You can imagine this on plants that are trying to deal with VOC loads or carbon sequestration or all sorts of things. You could imagine going after that.
And the ability to make these grid-like patterns, as you pointed out, increases the surface area to volume ratio. So it would increase the efficiency in a smaller space.
So for example, if you had an oil spill in the ocean, you could make a grid and just put it right in the oil, right?
Right.
And then when it's done, you pull it out and wash it, and you could reuse it maybe.
And if you think about it, with the spill that we had in the Gulf a few years ago, they were effectively just inefficiently dumping fertilizer into the major parts of the oil, and the microbes were already there, and it wasn't as efficient. Could you imagine if you had the right mix of oil-degrading organisms suspending in these grids? You could effectively create a wall as the oil would move with the currents. It would degrade in real time, simply as it would pass through this. And because of the three-dimensional geometry, you're getting all that catalyst that is effectively going to degrade it or transform it so that it's not as sticky or as heavy. And you can have various organisms at different levels that may be chewing at one aspect of the oil, one particular carbon fraction versus another. And so by demonstrating that this works for something as straightforward as phenol, but you can well imagine that the next experiments are to build these complex geometries with organisms that will have different predilection for one carbon source over the other. It's whether your kids like peas or broccoli. and oil is composed of both peas and broccoli. And you could actually sort them out based on the organisms you dynamically load into it.
So to borrow a phrase from the movie The Graduate, I've got one word for you. Flink.
Flink. That's right.
Flink. It's the way
I've got one word, Flink.
And that could be the title.
That's right. I like that. So the other thing they do is they take Acetobacter xylinum, which can make cellulose when exposed to oxygen. So they make a matrix containing that bacterium, and then they put it on a face mold. So they have a mold of a person's face, and they layer this on top of it, and the bacteria make cellulose. They can then wash out the ink constituents, and they leave behind a network of fibers. They call it nanofibrillated bacterial cellulose. So they say this could be, skin. This could be used as human skin.
I thought that was just amazing.
Right?
Wow.
You could almost imagine that if you could create this matrix of bacterial cellulose that you could then deposit kidney cells, could you make a real-time dialysis unit where you would just pass the blood, where you would allow the waste products to go through this filter to effectively take out to do effectively what a kidney does and just return it. It's a variation of peritoneal dialysis,
that's
but
cool.
with a real machine.
Yeah. I mean, what we should point out is that you can remove the bacteria, so they can mold this face on a face, and then they can remove the bacteria, and then if they had to, they could put it on your face if you had lost all your skin. But you can get rid of all the bacteria, which is important.
And they also point out that this is not far-fetched. They have cited a couple of reports where bacterial cellulose is already being used in the medical industry for tissue engineering.
Right. That's right.
You can read more if you pull this paper off the World Wide Web.
You can imagine all kinds of films and coatings, implants to avoid organ rejection. And they also talk about incorporating oxygen-producing bacteria in these gels. And that oxygen is needed for the xylinum production of cellulose. So you can have really big things made, right? Not just because the experimental one they made here is rather thin because it needed to be permeated with oxygen. But if you include oxygen-producing cyanobacteria, you can make it much thicker.
Wow.
And you just add a light, so you don't even have to feed it.
That's right.
It'll just photosynthesize.
Yeah, it's a human face. I have a cute little picture there of this human face with the bacteria making a cellulose film on top of it. Skin replacements, organ transplantation, any 3D shape you can imagine, you can make. So that's pretty cool.
It is. I'm a little puzzled by how you would control, what's the optimal amount of bacterial replication you'd want?
I guess that's the sort of thing you have to figure out, right?
Right. And it probably differs depending on your application and which bacterial strains you're using. But it is a living, dynamic system.
Yeah.
And the carbon source that you feed it.
That's right.
Right.
They say, we envision that long-term medical applications will benefit from the presented multi-material 3D printing process by locally deploying bacteria where needed. That's just so cool. Unprecedented functionalities, adding a new dimension to 3D printing. That's very cool. So if you lose your skin on part of your body, they could synthesize a new skin out of cellulose and put it back on, for example. And as Michael suggested, maybe whole organs one day. So this is pretty impressive, I would say. The potentialities or the potentials of this are really amazing.
Yeah. They also point out that the incredible diversity in the microbial world, since microbes live everywhere on the planet in so many different environments that their metabolic, kind of the metabolic tool chest that engineers would have to apply this technology is essentially unlimited. Now, I'm sure there are engineering issues that are going to need to be solved, but the potential is amazing.
Yeah.
Flink. One word.
Flink. What did you say? The future is Flink?
I have one word for you, Mrs. Robinson.
One word for you.
Yeah.
Was it Mrs. Robinson who gave the
No,
advice?
no, it was a guy. It was the guy standing at the pool who was telling Dustin Hoffman that.
Yeah,
Right.
that's it. All you have to do is Google. I have one word for you.
Yes.
Flink.
Plastics.
And it's not plastics, it's Flink.
That's so funny. It's Flink, yeah. In fact, if you search, you get the clip from The Graduate, a 1 minute clip where he's saying it. Oh, that's so funny. A young Dustin Hoffman. Anyway, I thought that was an impressive paper, even though most of it is not microbiology, but other things. But it's still very cool. And there's a nice picture in it as well. I don't think this is an open access article. I don't think it is. But
No, it is.
Is it?
It is. It's distributed under a Creative Commons Attribution Non-Commercial License.
Very good. I can even use the image then.
Yes.
Wonderful. I love when they do that. Thank you very much. Well, there you go. 3D printing of bacteria into functional, complex materials. Just ask for one under the Christmas tree or the Hanukkah candle.
The dreidel.
Isn't that cool?
You know, I developed back in the early 90s, I developed a vapor phase bioreactor that would take these VOCs, and it was based on membrane oxygenator, in which we would blow the air across the oxygenator, and the biofilm was effectively growing on these hollow fiber polypropylene tubes that had little tiny holes in them. So the gas would effectively come, and the bacteria were on the outside of the tube, if you will, and they were bathed in media. And the problem with, depending on which medium that we used, is that you could only have so much catalyst at the surface. And this solves the problem of putting catalyst in 3 dimensions, because that was always our limiting capability of how much catalyst we could have per surface area. And the cost of the membrane was very expensive. But this then, makes the whole device get much, much smaller. And we could feed that membrane oxygenator as much VOC as we could pump through it, and it would be all gone in real time. And it was incredible to see how well it worked. But the engineering limitation we had is we had no way to cheaply make more of the catalyst available short of buying more membrane oxygenators, which were very expensive. So this solves the problem. And, because you can use, you can make as many color inks as you'd like, which means different bacteria. You can go after very complex waste streams that can be degraded in one single pass, which is really code for what a kidney or a liver is really doing in the human body. We're constantly feeding ourselves complex waste streams that our liver and kidneys deal with in real time. Yeah. Just by passing blood across that cell matrix.
Neat stuff. Okay, we don't have any emails, and so I think, yes, Michele?
I have a little current events.
Oh, sure.
Today is December 14th when we're recording this, and so on Capitol Hill, our elected officials are trying to reconcile the House and Senate tax package. And one of the issues that we're concerned about in academia is whether or not graduate student stipends are going to be taxable. Currently, you're not taxed on the tuition waivers that many graduate students receive. Currently, the word is that that will not be included in the final bill. So we are hopeful, because that will really drive up the costs of graduate education.
Yeah, if they have to vote on it, that should go out. But I'm hoping it'll not pass, because it's a lousy bill either way.
Yeah, that's a bigger question.
But I don't know what's going to happen. That's politics, and we are microbiologists. All right. So
Right.
we don't have any email, but I'm going to have to give away a book next time to start getting some emails. And so this episode will be released on the 21st of December. We'll record another one on the 28th. So then we'll do a book giveaway. That'll be released on the 4th of January. So early next year, you could start trying to win another book and try your hand. This is TWiM 167. You can find it at asm.org/TWiM. You can find it at Apple Podcasts. And, of course, if you have a phone or a tablet and you listen to podcasts on an app, very easy to subscribe. And please do that so you get every episode. It also helps us if you subscribe. That helps our numbers. And if you like what we do, consider supporting us financially. Go to microbe.tv/contribute. Some of you may know that Patreon had a little issue in the past week or so. They were going to charge patrons a certain amount of money, and that caused us to lose quite a few patrons who were contributing to us. So they have rescinded that move, and so I hope those of you who left will come back. If everyone just gave us $1 a month, that's $12 a year. That's like 3 coffees. It would help us so much, and so consider doing that. For the holidays. Give us a holiday gift. We'd be very grateful for that. And, of course, send your questions and comments to twim@microbe.tv. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you.
Very nice explanation of a virus today on TWiM.
Well, I like to keep the melody clear, not get bogged down in so many of the details.
And also you benefit because you do teach, and it helps with that, right? As well.
Right. And I am just winding up teaching a first-year seminar called Current Topics in Microbiology. And I can't wait to incorporate this one on 3D printing of bacteria in next year's edition of that class.
That'll be great. They'll love it. They'll love it. Yeah. Yeah, that's cool. Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, Michele and Vincent.
And I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the American Society for Microbiology for their support of TWiM, Ray Ortega for his technical help. I want to thank Ronald Jenkins, who wrote the music you hear on TWiM. He's at ronaldjenkins.com. And I want to thank all the listeners, especially those who support us financially, but everyone for listening and being part of the team. And I hope everyone has a great holiday and a very good new year. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.